All right, good morning, you guys. It's a beautiful day. Happy Monday. So, what we're going to be doing today, I am recording this lecture now, just so you know. I do have a few announcements. Let me go to the top here. We will have some top hat questions in just a second, so you can start getting your top hat devices out. Today we're going to finish with cell signaling. I'm going to talk more about G protein coupled receptors, just briefly, and then I'm going to get into cell physiology. We're going to get into the regulation of cell volume, calcium regulation. We'll talk about pH regulation within the cell, and I really want to impress upon you that a lot of the channels and the transporters that we talked about last week, we're going to apply when we talk about cell physiology today. So I want to just connect that back to what we learned last week, and again, we're going to utilize that. We're also going to be talking about epithelial cells today, and on Wednesday, we're going to start with neurons. Now again, I sent out an announcement this morning. I want to make sure that everybody, this is what we talked about on Friday. Exam 1 is postponed until Wednesday, October 2nd. Just a reminder. I did send out an announcement today on that effect. Also, I did post the instructor PowerPoint presentations from last week and the recorded lecture from Friday. So if you have any trouble, the recorded lecture is actually still processing. It's only got about 5 more minutes, and it should, by the end of class period, it should be up and running. But if not, just shoot me an email today and let me know. Also, I have to say kudos to one of our students in this class, did a little digging and actually found an email to one of the authors that used to work in Robert Lefkowitz's lab. Remember I said I didn't know what GQ coupled receptors, how they were named? This student actually found the answer. Now, take it with a grain of salt. This was an email to another. Here, I'll show you the paper here. Let's see. Okay, so the paper, there's 2 authors here. One is Mel Simon, and the other is Michael Strathman. And this is an email from, it's actually an email from the Zhang lab to Mel Simon asking Michael Strathman, who named the GQ coupled receptors, how he did this. And here was his answer. This is Mel Simon's answer. GQ designation comes from Michael Strathman. He couldn't use the front end of the alphabet because those early letters were already reserved for the classes of subunits. The GZ designation had been taken, and GX seemed too obvious, and so GQ. So this is a better answer than I've ever been able to provide for students, just so you guys know. And anyway, going forward, this is the answer that I will use. It's closer than any other answer I've ever gotten. All right, so let's get out your top hat devices. Just a couple of questions this morning to remind us what we did last week. True or false? Some receptors act as ligand-gated ion channels. Is this true or false? It should be an easy one, but I want to revisit these terms. All right, we got 71 answers. Oh, we're up to 76 now. Give you about 10 more seconds. We'll have another question here in just a second, too. All right. We will have another question, so you'll have another opportunity in just a second. And the correct answer is true. Nice. Just about everybody got that right. 99% of you guys got that right. Okay, so you might not remember this though. Let me switch over to the document camera. These receptors are ion channels, nicotinic acetylcholine receptors, and the terminology for these receptors are called ionotropic. I actually wrote this down last time. This is a term that describes receptors that are ion channels. They're called ionotropic receptors. All right, but what I didn't give you, this is another term, metabotropic. Metabotropic. These are receptors that are G protein coupled receptors. So, ionotropic means receptors that are ion channels. Metabotropic means these are receptors that are G protein coupled. Okay? Just want to make sure that you guys know this term now. These two terms, ionotropic and metabotropic. Anybody need more time writing that down? All right, so let's go back to our top hat question. Go to the next question here. Which G protein coupled receptor increases the level of calcium within the cells? This is a lightning round. Give you about 30 seconds. This should be very easy for you. All right, we have about 80 people already that have answered. I want to make sure that everyone in this room gets a chance to answer. Still need to get those top hat quiz points in. Magna did a fabulous job of getting the problem set number 2 done this weekend. Thank you so much. Okay, 5 more seconds. Does anyone in this room need more time to answer this question? Raise your hand high. All right. So the correct answer, of course, is GQ. Sweet. Okay, so if this is a little confusing for you, let me know. That is the GQ coupled receptor. I do expect for you for the exam, and we will go over this in the review session again. I'll write it on the board. We'll go through GS, GI, which are really very similar in their pathway. GS actually stimulates adenylyl cyclase, where GI inhibits adenylyl cyclase. They both regulate the amount. They have effects on cyclic AMP within the cell, which is a second messenger. GQ coupled receptors, they release calcium into the cell. So let's start with the cell signaling. Go to our PowerPoint presentation here. Here's where we left off on Friday. All right, so on Friday we talked about phospholipase C. Well, let's go back and I'm just briefly going to talk about the GQ coupled receptor. If this isn't in your notes, this is the GQ coupled receptor right here. One very classic example of GQ coupled receptors are the purinergic receptors. They're activated by UTP and ATP. We'll talk about that when we get to epithelial cells. Once your ligand binds to the receptor, there's a conformational change that promotes the exchange of GDP for GTP. So at rest, the alpha subunit has a GDP attached to it, guanine diphosphate. It exchanges the GDP for GTP, and then it activates it. Now remember, the alpha subunit is a GTPase, so the clock is ticking. It's only a matter of time until that GTP is hydrolyzed back to GDP. But while it's activated, it will associate with phospholipase C. And I gave you the terms for PIP2 and IP3 and DAG on Friday, but really for our purposes and exam 1, you just need to know PIP2, phosphatidyl inositol bisphosphate. That's PIP2, and then IP3 is inositol phosphate, triphosphate, and DAG is diacylglycerol. So again, you don't need to know that for exam 1, but do know that PIP2 is converted within the membrane to IP3 and DAG. What I didn't tell you on Friday, because we ran out of time, DAG actually goes on to activate protein kinase C. Protein kinase C then goes on to activate many other proteins. Remember, a kinase is something that phosphorylates proteins. So uses ATP to phosphorylate and activate other proteins, and again, there's a global response. That's what diacylglycerol does, and it's actually membrane bound. Whereas IP3 enters into the cytoplasm, it has receptors on the smooth endoplasmic reticulum, the smooth ER. That's where all the calcium is housed. It releases some calcium, but then the calcium activates more IP3 receptors. It's kind of a calcium-induced calcium response. It is a positive feedback mechanism. We've talked about that before. It amplifies the response, so this is a global calcium response, and the cell is truly activated at that time. IP3 and DAG, these are second messengers, just like cyclic AMP with the GS and GI pathway. Please let me know if you're confused about this. You can, come into my office and talk about it. A lot of students are taking advantage of that. Or you can wait until the review session and I will go over this once again. All right, so let's talk now about, these are true cell physiology concepts. The first one I want to talk about is regulatory volume increase and decrease. This will be important when we get to epithelial cells, which I am going to actually apply to cystic fibrosis to help you with your homework assignment. I did notice I had a student email me on the Canvas website that is due on Friday. I think in class I told you it was due on Wednesday, but I'm going to stick to the Canvas website, so that will be due on Friday. Okay? All right, so now let's go on the very right-hand side here. This is what happens when cells start to swell. So when you drop it in a hypotonic solution, cells are going to start to swell. And in order to prevent lysis, the cells quickly adjust. They're trying to bring their cell volume back down to normal. And by doing that, they activate different channels and transporters. So they may activate a potassium channel. Potassium moves out of the cell. In this case, with this cell type, when this chloride channel opens, it actually moves chloride out of the cell. Remember, that's dependent on the concentration gradient and the membrane potential, whether it's going to move in and out. In this case, both of these solutes move out, and the thing to remember from this entire slide is that water is going to follow that solute movement. This is the take-home message with this entire slide. Water follows solute movement. So when you move these solutes out, water tends to follow. Again, this is a mechanism to help shrink cells in the face of pressure. If the cells drop it in a hypotonic solution, it's starting to swell. This is to try to bring the cell volume back down to normal. You also have a few co-transporters. You guys remember this as also a symporter. Talked about this on Friday. Moving potassium and chloride out, water will follow. Also, in addition to some antiporters or counter transporters, the sodium-calcium exchanger, in this case it depends on the stoichiometry. 2 sodium are moving out, 1 calcium moving in. So the net transfer is 1 solute out. Again, water will follow. Also, primary active transporters like the calcium ATPase will move calcium also out of the cell. And as you remember, the pump has 1 net solute, I'll say an osmolyte. That's probably more applicable to what we're talking about here. The net movement is 1 osmolyte out of the cell. So in this case, the pump is revved up and it increases the turnover rate, moving solute or osmolytes out. Water is going to follow. So again, take-home message is water is going to follow that solute movement. All right, how about the other side here? Now you've dropped the cell into a hypertonic solution. I always think about red blood cells in the kidney. As it travels in the inner medullary region of the kidney, it is a very hypertonic solution, 1,200 milliosmoles, whereas plasma is only about 300. Those red blood cells really shrink. And in order to try to regulate their volume, they have these mechanisms, right? Sometimes there are channels that open up, solute actually moves into the cell. And again, depending on the resting membrane potential, in this case, chloride is going to be moving into the cell as well. And water follows. Okay, you're going to hear me talk about this co-transporter a lot, especially when we get to the kidney. This is my favorite co-transporter. Seriously, this transporter moves a lot of solute with every cascade event, every conformational change. This is, and this isn't very original, this is the sodium potassium 2-chloride co-transporter. Okay? Now, it again moves a lot of solute into the cell. It's moving 4 different solutes, sodium, potassium, and 2-chloride. Now, when we get to the kidney, we're going to be talking about drugs that actually inhibit this co-transporter. Lasix is one of them. Anybody heard of Lasix? It's a powerful, powerful diuretic. Anybody know what a diuretic is? We'll learn more about this when we get to the kidney. It essentially, excuse my language here, it just makes you pee a lot, right? It's really to help you get rid of fluid. So, funny story, Lasix actually inhibits this co-transporter in the kidney, which is a diuretic. If you go to the horse races, did I tell you the story already? No. If you go to the horse races, the horses sometimes are given Lasix after a race because they want to get rid of tissue swelling, edema in their legs, or sometimes horses get really major nosebleeds, right? Racehorses do. So when that happens, they give them Lasix to try to get rid of their bodily fluids to mitigate those symptoms, right? So I thought I was really smart. I go to the horse races. I'm looking at the program, and I'm going to bet against all of the horses that are on Lasix because they tell you right in the program if they're on Lasix or not. And don't do that. I lost a lot of money doing that, actually. So, but that actually tells you, right? Lasix is a very important diuretic, and we'll talk a lot more about this when we get to the kidney and why that works. Okay, sodium potassium 2-chloride co-transporter. And then this one, I'm going to let you talk with your neighbors for a minute. Why would this be the case? We've talked about the sodium proton exchanger before. It moves 1 sodium in and 1 proton out. Why would this be a good exchanger, a good secondary active transporter for regulatory volume decrease? So I'll give you a couple minutes, talk to your neighbor, try to come up with a rationale why that would be effective. right, does anyone have an idea? Does anyone have an idea? Why would this be effective? So I heard a lot of students talking about pH, and that is true. The sodium proton exchanger is actually something that is going to have an effect on pH. But in this case, we're just talking about it as an osmolyte, right? So why would that be? What happens to protons sometimes? Protons tend to bind to what? What's that? Anions, proteins that have a negative charge, buffers, right? And if it binds with something like that, it doesn't act as an osmolyte. Okay, so this proton disappears if it binds to a buffer, and really the net movement is one sodium in. And that's going to cause, right? The water's going to follow that solute movement. All right, so let's go to the next one here. Calcium regulation. I'm not going to go into too much depth on this, but calcium is tightly regulated in the cell. Tightly regulated because it is what is initiating muscle contraction, whether we're talking about skeletal muscle, cardiac muscle. Once calcium floods into the intracellular space, muscle contraction that mechanical contraction can occur. So you can imagine that it has to be tightly regulated in the cell to really help with muscle, normal muscle processes. Okay? So here are some of the players. We have the calcium ATPase. This is a primary active transporter that moves calcium into those internal stores. This is the smooth ER. Now, when muscle is stimulated, it activates this ryanodine receptors. We're going to learn this next time. Calcium floods into the intracellular space. That's in pink here. And then once that muscle restores rest, it goes back to rest. These are the exchangers that are important. The sodium-calcium exchanger uses the sodium gradient to move calcium out of the cell, and another plasma membrane calcium ATPase. Other channels in the plasma membrane are the dihydropyridine receptors, again, very important in muscle contraction. So, just a sampling, just taking a look at some of the channels and secondary and primary active transporters that are important in calcium regulation. So we'll revisit this. I mean, you don't need to know or memorize any processes. I'm just giving you a sampling of what the players are. Okay. And finally, this is the last of the cell signaling. This is an important concept. Yes, the sodium proton exchanger is very important in pH regulation. This is supposed to simulate some kind of cell here, this square. And inside every cell, you're going to see that there's many different buffers, including bicarbonate, phosphates, and proteins. Now, I just want to remind you, I know that you all know this, but pH is a measure of free proton levels. pH is a measure of free proton levels. So these buffers inside the cell are going to help to stabilize pH, but once you've overwhelmed these buffers, you're going to see some dramatic changes in pH. And these are secondary active transporters that are really important in keeping pH in a very specific range. So normally the pH of intracellular pH of cells is 7.2. So if the inside of the cell becomes more acidic, let's follow this line down. What that means is there's an increase in free proton levels. That's why we have a plus, H plus. An increase in free proton levels and the pH starts to fall. When that happens, pH has effects on the sodium proton exchanger. And like we've talked about before, I hope you can visualize this. It uses the sodium gradient, brings in sodium from outside to in, to remove free protons from the inside of the cell. To remove free protons from the inside of the cell. So it uses the sodium gradient to remove free protons from inside of the cell. Once you remove those free protons, intracellular pH actually increases back into normal range, about 7.2. So that's how you read this. You're going to get an increase in the sodium proton exchange activity, that exchanger's activity. So let's go in the opposite direction. What happens when the pH increases and falls out of range? That's what this negative. What's happening is free proton levels are decreasing. So it makes sense that you're going to shut off or decrease the activity of the sodium proton exchanger. So first thing you're going to do is shut off. So you're going to start to accumulate some of these. But also what's happening is you have a chloride bicarbonate exchanger. And what you're going to do is remove your buffer. It's going to use the chloride gradient to remove some of your buffer, which is also going to lower the pH. You're going to start to accumulate more free proton levels, You're going to increase those free proton levels if you remove your buffer. So, going back to, you're going to increase your chloride bicarbonate exchanger's activity, removing your buffer so that you can bring the pH back down to normal levels. Any questions? Does that make sense? Let me know if that is confusing. I do have a lot of students that come up after class and ask, can I go through this again? But what we're doing here is we're applying our exchangers, our ion channels, and those are the facilitated diffusion mechanisms, and our active transporters, whether that's primary, secondary active transporters, to a physiological process. Okay, so let's talk about epithelial cells, and this is going to be very important in basically the rest of the semester when we talk about kidney and GI tract, and it's really important to know what epithelial cells do, and this is going to be important in helping you with your homework assignment that's due on Friday. All right. Epithelial cells are it's all about transport. All right. So what epithelial cells are they are cell types that line your different organs. They are the barrier between the outside world and the inside of your body. The barrier between the outside world and the inside of your body. And so whether we're talking about the lining of your lungs, the lining of your intestines, uterus, right? Again, these are structures that are constantly They are continuous with the outside environment. So these tissues also have physiological functions such as respiration, digestion, and ion and osmoregulation. That's what we're going to be talking about with the kidney. There's different types of epithelial cell structures. Some are flat sheets like the epidermis. The epidermis really doesn't have a whole lot of transport properties, but it is a barrier. Kidneys are organized in what's called tubules. So let's take a look at that in this next slide. I'll come back. These are tubules. These are the epithelial cells here, and they are what's considered polarized. I'll talk about that in just a second. They have different proteins that are sorted to either the luminal side, that's the interior of the tube, or the basolateral side faces the blood supply. It's also called the serosal side. So they're organized in such a way that they have transport properties that can modify the inside of the fluid within the lumen. They can modify the fluid inside the lumen. So you can see the tubules here, just tubes, like the intestines as well. And then there's glands. And I think this is going to be important with cystic fibrosis. We're going to talk about acinar glands in just a second. The simplest gland type are the crypts in the small intestines and colon. Okay, we'll start with that. It's just a U-shaped structure that does secrete a lot of fluid to help lubricate the intestines, to help with digestion and movement within the intestines. I'm going to concentrate on these acinar glands today. Those include the salivary glands, mammary glands, pancreatic glands, those exocrine glands, and sweat glands. And we'll talk about that with cystic fibrosis. So those are all types of acinar glands. Number 2 here is a typical acinar gland. It has two structures. One is a bulb-like structure where transport occurs, usually from blood. It moves fluid into this acinus, and the fluid is very plasma-like at first. But then as that fluid moves through the duct portion, this is the duct, that's where the fluid gets modified. With sweat glands, this is where CFTR is expressed, in the duct cells. Now, if you remember back to Friday when I showed you the video of Spencer, the little boy that has cystic fibrosis, he had salt deposits on his head, and we'll talk about why. And then tubular glands like endometrial glands. This is a typical endometrial gland in the uterus. They're very complex glands. They can be branched or coiled. All right, so going back to the acinus, I want to just give you, when we get to the hormone section, I'll talk about the difference between exocrine glands and endocrine glands. So when we're talking about these exocrine glands like the pancreas, sweat duct, mammary gland, again, here's a typical acinar gland. This is the acinus and duct portion. These are called exocrine glands because they deliver molecules to the external environment, like the skin or the inside of the small intestines. Whereas endocrine glands, they deliver their molecules like hormones directly into the blood supply to be circulated throughout the entire body. So some terminology that's really important. And again, there's a lot of terms on exam 1. There are different occluding junctions. So these are two epithelial cells that they've come in contact with each other. And as soon as that happens, they actually form what's called tight junctions. So you can see that they're actually forming junctions, communicating with each other, gives them the capability now of sorting different proteins to different sides. This is called polarization. Desmosomes, these are called spot welds. They're a little bit stronger in their interaction. They've got these cytoskeletal filaments that are interacting with these glycoprotein filaments. Again, it's just a tighter, more stable interaction between the two cells. And then finally, these are called gap junctions. I'm going to spend a little time talking about gap junctions. These are interactions between two cells, just like you see here, but in this case, they form a channel. They both are expressing what's called hemichannels that interact with each other to form a complete pore region. To help with the movement of calcium, monovalents like sodium and potassium, it allows the movement of these intracellular electrolytes to move back and forth from cell to cell. This is really important in cardiac muscle. The fact that a wave of depolarization can go from cell to cell to cell so that you can get a coordinated heartbeat is really important. You want your entire heart to beat at the same time to propel blood out of that organ. So gap junctions are very important in that process. They're specialized protein complexes that create an aqueous pore between two adjacent cells, which allows for different chemical messengers to go from cell to cell without ever having to go into the extracellular environment. And again, it typically involves movement of ions, which is responsible for the depolarization wave to propagate through the entire heart to again give you a coordinated heartbeat. So each one of these connexins is a subunit. There's 6 of them. It forms a hexamer. This hexamer is called a hemichannel. And then 2 hemichannels come together to form a complete pore. Again, these are called gap junctions. So a couple of other terms. Whenever molecules like sodium or chloride move through the cell, right, they come in through a channel or a transporter and they exit through a channel or a transporter. This is called the transcellular pathway. And then, if that sets up a voltage gradient for the movement of other electrolytes in between, sometimes those tight junctions allow for electrolytes to move in between cells. This is called the paracellular pathway. Para means next to, next to. So it's basically the electrolyte is moving next to each of the cells, paracellular. So let's take a real-life example. A real-life example, I'm going to show you how epithelial cells work. Again, you can apply this to your cystic fibrosis homework. You can isolate glands because they're actually very dense and heavy. You can put them in a centrifuge, and you can see this is a electron micrograph of, or just a micrograph of this epithelial cell gland. It's isolated from the uterus, so it's called endometrial glandular epithelial cells. Centrifuge it out, and this is what they look like. Under the microscope, they still have their 3-dimensional structure. And if you put them in a tissue culture dish, cells will grow out radially from this structure until they proliferate into what's called a monolayer. Okay, so these are glandular epithelial cells. Once they monolayer, you can use an ohm meter to measure their resistance as they grow in and pack in and form tight junctions. the ability for electrolytes to move across the entire monolayer gets more and more difficult. So the resistance goes up, and the oscillation just reflects media changes. When you change the media, it slightly goes down, and then after a few days, it goes back up, right? So that's why it oscillates, but it pretty much keeps its resistance. That's how you know that it's completely monolayered. So when the cell forms those tight junctions, right? You can see that here. That's when it starts to sort different proteins either to the apical side. See all these green ones now on the apical side. They're facing the lumen, or the external environment, or the basolateral side, which is facing the blood supply. All these pink dots are now being sorted to the basolateral side. Now you can promote vectoral movement, one-way direction of movement of electrolytes. So again, let me just introduce the idea of polarization. Once they form tight junctions, epithelial cells become polarized. This is kind of a crazy slide here. Don't be scared. I'll walk you through it. Oh, it stays on that slide. All right, so, oops, let's go back. There it is. Okay, so can anybody tell me what pathway this is? This is a metabotropic receptor, G protein coupled receptor. What pathway? You can just, yes, GQ. This is a typical GQ coupled receptor. It is a purinergic receptor called P2Y4. Again, you don't need to know the details. I'm just going to give you an overview of how epithelial cells work. Once this UTP, which is the ligand, binds to its receptor, it activates the GQ pathway, releasing calcium into the intracellular space. In this case, PKC, remember DAG is going to activate PKC. It activates these chloride channels. Calcium activates the calcium activated chloride channel, and PKC activates actually CFTR. And my favorite co-transporter, the sodium potassium 2-chloride co-transporter, is actually loading chloride into the cell. So what you have here is vectoral movement of chloride through the transcellular pathway into a gland. Let's draw this out. That looks pretty complicated. Let's draw this out, actually. I think you'll appreciate this. Here is a typical gland, like an endometrial gland. It's a little more complicated, but I'm going to draw. These are my cells. This is a monolayer of epithelial cells. Right? And we have our nucleus in each one of these cells. Right? Okay, so what's happening when I activate the GQ coupled receptor and calcium is mobilized into the cell? It promotes the transcellular movement of chloride into the interior of the gland. That sets up a voltage gradient, right? I'm going to put this as a voltage gradient, where the inside of the cell is more negative than the outside, right? Because you're moving all of this chloride into the cell, into the gland. It sets up a voltage gradient, which then promotes the movement of sodium to follow the chloride movement into the interior of the gland. And it's going across the pericellular pathway in between cells. Which way is the water going to follow? Yeah, it's going to go in with the solute movement. So water's going to go into the interior, both through the transcellular and pericellular pathway. So what, I should say it this way, the solute movement increases the osmotic driving force for the movement of water to follow. You may not have realized how your glands work. It's not by magic, it's actually by biophysics, right? Moving solute in, water follows. This helps to deliver water, nutrients into, in this case with endometrial cells, into the uterus to help with concepsus viability. All right, so we only have a few more minutes. Let me just show you how sweat glands work. And then if this will help you with your homework assignment, this is a typical acinar gland. This is the acinus portion. Okay, so fluid from blood supply is very plasma-like. CFTR is expressed on the apical membrane, right? And what usually happens is the fluid moves into the duct portion and chloride is transported out of the duct along with sets up a movement of sodium. But water in this case isn't allowed to follow because there's not as many water channels that are expressed in the duct portion. And again, CFTR is responsible for this reabsorption of salt. And usually the fluid that is produced is very hypotonic compared to plasma. But if CFTR isn't present in the membrane, you can see what happens then is salt is lost along with water on the skin. That's why Spencer actually had those salt deposits on his head. So I'm going to let you struggle a little bit with the lung epithelial cells. That's really what your homework assignment is focusing on, but we will revisit this. So try your best to figure out what's happening in the lung, and we'll talk about it in class on Friday. All right, I'll see you on Wednesday. We're going to start with neurons and nerves on Wednesday.